Okay, welcome to the recording that will help you out using NCBI and BLAST technology in order to develop genetic trees for phylogeny. So you've already worked with your sedges and doing morphological trees. Now we're going to apply genetics to it and look to see how they compare, and this will give you the push you need for where you're going in your research on genus Carex. So first thing you're going to do is you're going to pull up Google, type NCBI just like I have here, and you're going to look for National Center for Biotechnology Information. So we click here, and it pulls up this beautiful website. Notice that there's a red bar here because of the government shutdown. That's not going to pertain to us necessarily, so keep that in mind, that all your information that we're going to be looking at is currently up to date. So first thing you're going to do for your first piece is to look at how different species compare. And one of the first things that you're going to look at for species comparison is a homologous gene. So homologous means similar, and we're going to be looking at an enzyme that is common to organisms that do glycolysis, which is pretty much every single organism on the planet. So you're going to look and click on all databases and change that to homologene, and we're going to type in the enzyme. So that enzyme is GAPDH, which stands for glycerolaldehyde phosphate dehydrogenase. So here it is right here. For results, we're going to click on that, and that should pull up our different organisms that they have already done for us. So you can see here is us, Homo sapien, and then there's all these other ones that you're going to have to do a little bit of research and figure out what they are. I'm not going to unveil that for you yet. So it pulls up all this information. Now you have the ability with display settings to be able to look at different types of information that scientists have already put in here and compared computer-wise. So one of the things we're going to look at is how the alignment scores look. Apply that. And it pulls up, one of the big things here is proteins, right? So glycerolaldehyde phosphate dehydrogenase is an enzyme, so it's a protein, 3D conformational shape, very specific. And for Homo sapiens compared to this organism, P. troglodytes, which is a chimpanzee, 100% of that protein for glycolysis is exactly the same. 99.6% of the DNA that is coding for that protein is exactly the same. Here's another organism, and we continue down the list. Now, where we have multiple organisms that are exactly the same, they just took 3 different samples. So we have comparisons within the species, and we have comparisons across different species. So we're going to go back now. After you analyze that, you can look at comparisons in your list there. We're going to go back to our homologene, and we're going to get the download for our file. So we're going to click on download. It brings up this piece. They say download. What do you want to download? We're going to download just the protein, and it's going to give us this FASTA format. That's what we want for all of our analysis. So we're going to select all, and we're going to download. Right? I've already marked all those, and something should pop up on your server that says, do you want to save or open this? I'm just going to open it. Obviously, you're going to have to save it, and it pulls up this Notepad file with all this chaos. Okay? All you really need to know at this point is that you have to get it in a usable form. So one of the things that you're going to have to do is you're going to have to transform this file from just saying all this code into a name that makes sense, right? So one of the things that you're going to change it to say is whatever Homo sapien boils down to be. So when you look this up, maybe you're going to have to Google it. Homo sapien is obviously a human, right? And we're going to alter the entire rest of this format. And if you read your protocol for this activity, when we rename this, we want to bring it back to just the beginning piece. So we're going to block out all of this here and bring it down to just say Homo sapien. We're going to keep all the rest of this. We have to do that for every single file. So eventually, your final file will look something like this. right? So we have human as the alter and chimp and whatnot. And this will be your Notepad file that you're going to then save to your desktop or wherever it is that you're working. And then we're going to use that file to import into our next website. I'm going to open up Google again. I'm going to show you what we're going to be looking for. The website is called Clustal Omega. It is a free website that is going to be a multiple sequence alignment tool, allow us to generate our phylogenetic trees. Easiest thing to do then here is just browse for your file. So we saved it. I saved mine on the desktop. You find that desktop file, we import it in, and then we just submit. And depending on how many people are using it or where you're working with your server, this could take a couple minutes. Mine here, it just clicked through within a couple seconds. And we're going to be able to use this now to get all kinds of different things that we need. So here is the raw data of comparison. We're going to do a results summary that gives us a little bit more information. Not pulling it up right now. Oh, we have to have a child view. Phylogenetic tree is where we want. You pull it down. Down here is your cladogram that you're seeing. Now we can take different things and we can change showing the distances on the tree, which will give us a little bit better understanding of which ones are closer related than others. But this is what you're going to need to use for your final paper that you're putting together, your poster presentation. Obviously, you're not going to be comparing glycerol, aldehyde, phosphate, dehydrogenase. You're going to be comparing a different common gene found within Carex genus. So here we have my tree that I've put together. It's not necessarily correct. I just threw it together so that you have a comparison. So when you go back to this database, when you're doing your sedges, you're going to be looking for a different kind of information. So what we're going to be looking at is the nucleotide sequence database. So down here, we're not going to do homologene, we're going to do nucleotide sequence. And we're going to be looking for external transcribed spacer, which is something that the plants use to generate proteins. It's a common sequence that's used by a lot of scientists to compare between species and within species, creating very, very good trees for comparison. So you hit the nucleotide piece. We're then going to type in the different species that you want to look into. So maybe yours is Carex abrupta. Hit enter, and it should pull up. Oh, no results found. So let's see if we can fix. Okay, sorry about that. So make sure you type it in right. So when you hit nucleotide and then you hit Carex abrupta, pulls up two different ones here. One of them says internal transcribe spacer, which we're not going to use. We're going to use external transcribe spacer. Dr. Hepp said that this is a much more valid piece to use for our tree making. And you can actually see Dr. Hepp was the one that actually uploaded this information for us. So when we click on this, it'll pull up a bunch of information, but really all we want is the FASTA file. Because remember that Clustal Omega is going to use that FASTA file. So let's click on that. And when it does that, it pulls up this code. And all we're going to do is we're going to copy this code and we're going to paste it. So Control C, those of you that are using Windows. And we're going to paste it into just a blank note file. All right, so there's our first one for Carex abrupta. Remember, we can delete all the information that's before this little triangle. Right? Try to keep out the spaces, and you should have Carex abrupta, voucher hip, right? Hit a space, and then we're going to put in our next species in here. Now, I would definitely suggest putting in at least the 4 that you analyze in your scatter plot, and then pick maybe 3 or 4 other ones from your list in your database that was collected both by you, your lab partners, and other young scientists into this database that you have access to. I believe your teacher should have made a link for that on your Blackboard file for all of the Carex species that we're analyzing. So maybe another one that you're analyzing is Carex, let's see, Christella. And Christella has multiple different things put in here. The one that you want, obviously, again, is to compare to the common piece that we're looking at. By the way, we'll talk about ribulose biphosphate later on this year. It's a good one. It's called Ribisco for short. It's going to be one of our CO2 fixation agents in the Calvin cycle. But we need external transcribed spacers. So as I keep going down here we see Carex Christella, external transcribed spacer. Here's a complete sequence. You're going to hit the FASTA file for that. Remember, we don't want internal transcribed. We want external. We literally just copy and paste this into our note file. Right? Make sure that you rename it. Get rid of this stuff. And the only reason we rename it, because if you end up throwing this through Cluster Omega website, what it'll end up doing is punching out a chart, a phylogenetic tree, that instead of having the actual name that you want for information, it'll end up putting out this code piece on the front half of this. And if you didn't memorize it or write it down, then it makes no sense to you for comparison between species or within species. So once again, you need to run NCBI for a homologene complex between a lot of different species that are using glycerol aldehyde phosphate dehydrogenase, which is this common enzyme that's used in glycolysis, which we'll talk about, I believe, in chapter 9 of your textbook. And then we're going to use that same concept of using a common gene or common protein to analyze between species for our sedges or our research project that we're doing. So now, by the time you finish this, you'll have a printout of a genetic tree for your different species within sedges, and you'll have a morphological tree that you and your group have created. You'll have scatter plots to support this information, and then there's databases that are on Blackboard that we've loaded up into your Carex research folder. Two of them that were distributed to us from Dr. Hip and Mrs. Hahn that'll help us get the directions maybe that you want to go as a group. We're going to be comparing these for ecological purposes, climate, distribution, globally, evolution-based, but you're going to have to come up with conclusions on your own. Remember, there are seven questions on the back of your scatter plot analysis packets that will help you hone in on where you are and where you need to go before Dr. Hip comes in to talk with you in the next week or so. If you have any questions, feel free to email me, and otherwise, good luck.